Okay. Good afternoon, one and all. A warm welcome to this highly anticipated creative online talk on drug design. It is truly privilege to have you all gathered here for 114th episode of Creative Online Talks. This webinar is not just an opportunity to expand your knowledge. It's a chance to be a part of ongoing revolution in the medical field. Creative. Creative is a group of people who are eager to gain new knowledge. Creative, Cre means creativity, active means activeness. To be active, we need to be creative. So creative tagline is knowledge square, sharing of knowledge increases our knowledge. Vision of creative is to build constructive thinking on various domains. Our main focus on non-textual, non-academic, non-syllabus related concepts. Creative is thankful to all our resource person for engaging us with their knowledge. Every Saturday, we organize online weekend talks on Zoom platform and we live stream on our YouTube channel, Creative GBD. And we encourage each one of you to participate actively and ask questions at the end of the session. To present today's session, we have Mr. Kiran Bettapura with us today. Welcome you, sir, and a brief introduction about today's speaker. Kiran sir brought up in South Bangalore, got a bachelor degree in chemistry and biology from St. Joseph College in 2008, followed by a master degree in chemistry in 2010. Worked as a chemistry lecturer for 2 years before joining the Indian Institute of Science. Got a PhD in organic chemistry in 2018 and spent some time in USA as a postdoctoral researcher in a neuroscience lab, working as a rare neurological disorder. Came back to Bangalore and took up a job as a drug designer at BBMSRC. We welcome you, sir, and I welcome all the participants for today's session. Over to you, sir.
Thank you, Supriya. Can you hear me now?
Yes, sir.
Okay, I'm going to go ahead and share my screen.
Okay.
You should be seeing it.
Yes, sir. Visible, sir.
Okay, thank you. All right. So good afternoon, everyone. It's a pleasure to be here. I always like to talk, so this is a good opportunity for me to expose people on, to give some exposure to people about what drug design means. Usually, drug design has a lot of negative publicity associated with it because the term drug means a lot of things. Let me clarify in the beginning what I mean by a drug before we start getting into the details. So what is a drug? So all of us essentially use it in some form or the other. It is something that alters the way your body works, right? It can be beneficial for you, in which case it's usually called a medicine. It can be harmful or it can be recreational, right? All medicines that we consume, whether it's paracetamol or an anti-cancer agent, whatever we consume, they're all drugs. A lot of natural products that we consume, like the caffeine in coffee, is a drug. Paracetamol that we consume through Dolo 650 is also a drug. So drugs can be natural or they can be man-made. Like I said, you can have good things like caffeine is good for certain people, so you consume it. Paracetamol is definitely useful for a lot of people. Cocaine may not be useful for everyone, but some people use it for recreation. And those are the sort of things which can be eventually harmful. But at the end of the day, all drug substances around us is a chemical, right? It's basically a very simple organic molecule that can be made in the lab. That does not mean that all drugs that are sold or all drugs that are available have to be man-made. They can be natural. They can be semi-synthetic. By semi-synthetic, I mean there is an already existing starting material that's available in nature and that is harvested and converted into a drug, or it can be completely man-made. So all three varieties of drugs are available. But what you want to take home from this is anything that alters the way your body functions is a drug. If you have a disease, if it helps you come out of the disease, it's a drug. If your body is normal and it upsets the way your body works, it's still a drug, right? So let's think about what happens whenever we consume a drug, right? Most of us consume drugs usually in the form of a tablet. Young children usually consume it in the form of a syrup. Adults also do consume some things in the form of syrup. There are advantages of a syrup, and there are also disadvantages of a syrup. So let's say you consume a drug tablet, let's say paracetamol. So think about what happens to that tablet as you swallow it. Usual recommendation is you swallow the tablet with a bit of water, right? So that the tablet dissolves quickly in your stomach. So when the drug goes into your stomach, it disintegrates. Not the drug disintegrating, the tablet disintegrates, right? When the tablet disintegrates, it allows the drug to be exposed to the fluids that's there in your stomach or in the intestine. Now, the first property of a drug that's taken orally in this form should be a scenario where it should not degrade on exposure to stomach acid. Only then it can become a drug that is oral, right? If it is not consumed orally, then it can be consumed in other ways. We'll talk about that later. But most drugs that are consumed orally have to be resistant to the pH that's in the stomach. So the tablet goes into the stomach, it degrades, it breaks down. And as it breaks down, the tablet starts dissolving in the gastric fluid. And then it moves into the intestine. Once it comes into the intestine, now in all the gastric cavities or in the intestinal cavity, there is no blood, right? There is no blood that's exposed to food. There is no blood that's exposed to your drug. So the drug is still kept away from blood because of the lumen that you have inside. So now, after the dissolved material comes in contact with the intestinal walls have cells which are capable of absorbing the food or, for that matter, the drug. So now, if your drug does not get absorbed by the intestine, it gets wasted because it will just pass through the intestine, come out of feces, and nothing gets absorbed into your body. However, there are drugs that are administered which may not really be amenable to oral dosing, and they may be better administered through routes like intravenous, which is directly into your bloodstream, right? So when you get admitted to a hospital, rarely does the doctor ask you to swallow the drug. Most of the time, the doctor will ask you, lie down, they will inject it directly into your bloodstream, and that is intravenous. Or they can do it by inhalation, or you can have lingual absorption, sublingual absorption, basically below your tongue. A lot of nicotine addicts consume nicotine gum, chewing gum, basically. As and when you chew that gum, nicotine gets released, and that nicotine gets absorbed in the tongue, in your oral cavity. So that's another way of dosing yourself with a drug. There's also things like how you inject a drug into your muscle. Sometimes when you go, you get vaccine shots on your arms, or drug shots on your arms. So those are intramuscular ways of delivering. So the drug and the intended target of the drug basically decide how it's dosed to the body. It's very convenient to have an oral drug, but the limitation for an oral drug is it should be absorbed in the intestine. And it's a key step in that whole process. Now think about what happens after it gets absorbed onto the intestine. So on the other side of the intestine, after the absorption, is blood, essentially. There are lots of blood vessels around the intestine. The blood moves from the lumen, that is the gut, through the intestinal cell and gets into blood. And from that blood, it gets taken to the liver. And the liver is a critical organ for your body. It's the first checkpoint, really, a very important checkpoint for all your drugs. So your drugs have to clear the liver, have to pass through the liver. And when they pass through the liver, they get to interact with the enzymes that are present in liver. There are lots of enzymes in the liver, and their primary job is to break down molecules that are not necessary for you. So think about how living systems have evolved over so many thousands and thousands of years. So the liver has basically evolved to pick out things that are not necessary for you and then degrade everything that is unnecessary. So a lot of the drugs that you consume go through the liver and they are likely to get degraded or destroyed in the liver. Fine. Let's say your drug does pass through. We'll talk a little bit about how the drugs pass through the liver in a while. Let's say your drug does pass through the liver, and then it's now free to enter blood circulation. So once it enters blood circulation, it's now starts getting distributed all over the body. After it crosses the liver, there is no barrier that's preventing the drug from entering everywhere. Wherever the blood flows, your drug will flow. The only exception being the brain, because the brain has what is called a blood-brain barrier. Not all drugs pass through the brain. There are some drugs have to have certain features. Only then they're able to pass through the blood-brain barrier and reach the brain. So for all practical purposes, you can in general say anytime you consume a drug, if it gets absorbed at the intestine, if it passes the liver, it is going to get distributed to every single part of your body. Wherever there is blood, the drug goes there. That's the usual way it works, right? So once it's now distributed all over the body, it can now go and act on your target organ. Let's say you took a tablet to fix an issue with your heart. Now the blood is obviously circulating through the heart to help the heart muscles get some energy. The blood has to keep circulating through the heart. So the drug is carried to the heart. And as it gets to the heart, it finds and goes into the cells. And once it goes into the cells, there are like thousands and thousands of proteins inside these cells. And the drug goes and binds into those cells, finds the protein that it was designed for, and acts on the protein. So until this point, it's actually a very, very complicated process. You took the drug in a form which was in a tablet, and then ultimately it goes and finds the target protein that it wants to find. It's not trivial the way it happens. So I'm going to walk you through a bit of what happens in that process today. So the drug has to travel through all the organs, right? And in all the organs, it gets exposed to several different proteins, but it has to bind to only one protein, which it was originally designed for. And once it binds that protein is usually called the target protein. Now, however, after all this happens, you have to also realize that the drug cannot stay in the body forever, because the longer the drug stays in your body, the more chances of it having off-target issues. So off-target is essentially where your drug molecule now binds to proteins that it was not intended to bind. So you wanted it to bind to a particular protein in the heart, but it binds now to a certain other protein, maybe in your leg, right? So you don't want that to happen. And that probability increases as more and more amount of drug stays back in the body for a longer period of time. So ultimately the drug has to be removed from your body. So this comes with two things in it. If the drug gets removed from the body very quickly, then there is not enough drug to carry out the therapy, right? If the drug stays for too long, then it will lead to toxicology. So all these have to be balanced, and that's where the whole design aspect comes in. So I'm going to walk you through how the drug actually gets absorbed in the intestine. It's slightly complicated. I'm not going to go too much into the details. I understand this crowd is mostly a general crowd, but if you know a little bit of high school science, you will be able to appreciate what's going on right now. Let me switch on the pointer. You should be able to see my pointer. Okay. So first of all, you think about why would drugs have to be consumed orally, right? Consuming an oral drug is very convenient because you don't need to be skilled at injecting yourself. Imagine if you had to dose yourself into your blood vessel. How are you going to find it, and how complicated it would be, and how easy it is to complicate the whole process by stabbing yourself the wrong way, right? So you don't need any expertise to consume an oral drug. That's why most drugs are preferred when they are dosed orally. It also leads to higher compliance because it is easier to consume. If you tell the patient you have to take this drug twice a day, morning once and evening once, it's easier for them to comply. If you instead tell them, no, you have to stab yourself in the arm every day once in the morning and every day in the evening once, they may do it the first few days and then they may realize, oh, this is very frustrating and painful, and they may stop doing it, right? So you get higher patient compliance if you also have the drug that's consumed orally. Now, like I said, absorption in the intestine is critical as long as the drug has to be dosed orally. So whenever you get a drug in the market, if the drug usually comes actually in a cardboard box, and inside that cardboard box is usually a sheet of paper, which is called a package insert. And that's usually information for the patient. And if you open that insert and you get a chance to read it, there will be a term called bioavailability. Usually it will be there. It's denoted by %F, and it tells you roughly how much of the drug actually gets absorbed in the intestine. Very rarely 100% of the drug is absorbed, but there are drugs that are highly absorbed, like paracetamol is a highly absorbed drug. Its bioavailability will be 90% plus. There are drugs which will be very low bioavailability. That means you consume a lot of the drug, but very little actually enters your intestine. So the bioavailability term is usually used to denote how much potentially gets absorbed in your intestine. Now, think about your intestine now. It's really a messy place to be in, right? The entire intestine is coated with a mucus layer, right? And then there is a lot of food that you ate. And along with that food that you ate, you have these drug molecules. Now, these the intestinal wall essentially is lined with cells. These cells are called enterocytes, right? Enterocyte. So enterocyte's job is to basically decide, this is food, I need this. This is amino acid, I need this. This is glucose, I need this. This is sodium, I need this. This is chloride, I need this. This is drug molecule, I don't recognize this, I don't need it, right? That's the job of the intestinal wall. The enterocyte decides what enters and what doesn't enter. But you can smuggle some drugs through, right? You can smuggle because the intestinal wall is not 100% tight. Lots of places have tight junctions, but there are also places where it's not very tight, through which drug molecules can permeate through. They can pass through and finally reach the blood supply. Okay, so this type of transport is called a paracellular transport. It's between cells. It usually happens if the drug molecule is really, really small, and if it is charged, usually paracellular transport occurs. Most often, drug molecules are larger than paracellular requirements. You will usually get something called a transcellular transport. So something that you would have studied a long time ago in high school would be diffusion. So drug concentration is higher on this side. Some amount of the drug permeates in, diffuses in, and that's called a transcellular transport. Sometimes this transport is aided by transporters, which may pick up certain drugs, right? Your body may mistake the drug as something that's essential and then pick it up. There are sometimes the cells also express what is called an efflux. So influx is coming into the enterocyte, efflux is going out of the enterocyte. So whenever a drug gets recognized by the efflux, whatever came into the cell realizes, oh, this is the wrong thing that I picked up, and it throws it out by efflux. So a lot of drugs have this problem of being recognized by efflux transporters or by efflux pumps. So you ingest a drug, it goes in, some amount of it goes through transcellular transport. Some amount may get kicked back out into the lumen, into the intestine by efflux. So those are the reasons why not 100% of the drug gets absorbed. There's a lot more science in how to design to improve transcellular transport, how to get rid of efflux problems. All those things can be sorted out, and that's where the design comes in. So what happens after it gets absorbed in the intestine? Like I said earlier, it moves to the liver. There's a hepatic portal vein. that carries whatever drug molecules got absorbed. It moves it into the liver, right? So you have to realize whether all molecules get absorbed in the intestine. No, right? Some molecules that are lipophilic. Lipophilic is lipid-loving. Molecules have to be sufficiently non-polar to get absorbed. But I told you earlier that your tablet has to dissolve in the intestinal fluids and the gastric fluid. For that, the tablet has the component in the tablet, the medicine in the tablet has to be sufficiently water-loving. So it needs to be some amount of lipid-loving and some amount of water-loving. And it also has to escape the efflux transporters. And you need to learn how to balance all those three parameters. And that's where drug design comes in. Now, why should something be lipid-loving if it has to be absorbed by the cell? Just recollect that cell membranes basically are a lipid bilayer. They're called phospholipid bilayers. So the barrier between the intestine outside, cell membrane in the middle, and inside of the cell, in this barrier, you have essentially lipophilicity, essentially lipids. So the drug molecule has to be sufficiently lipophilic to come into the cell membrane and then come out of the other side. But at the same time, it also has to be water soluble. So there's a fine balance that you need of how you design your drug molecules so that it gets absorbed. After absorption, we can take a look at what happens in the liver. The liver, like I said, is a critical organ that protects your body from a lot of harmful substances. Every time you walk on the roads in a very dusty street, or you inhale diesel vapors or petrol vapors or some smoke, you need to think about what happens to all those things that you inhaled. How does your body get rid of it? A lot of it doesn't get absorbed in the lung, which is fine, but a little bit of it will get absorbed in the lung. And once it gets absorbed in the lung, it ultimately travels to the liver where it gets broken down. So the liver basically protects you from a lot of harmful material. So keeping your liver healthy is something very essential for you to lead a good life. If your liver has trouble, that means you have trouble maintaining your body in the state that it should be. Right now, specifically when it comes to drug molecules, molecules have to go into the liver. So the liver has a very simple job. Now, again, this is a function that has evolved over thousands of years. The liver knows what your body needs and what your body does not need. If anything that is non-polar goes into the liver enzymes basically work on those non-polar materials, convert them into polar materials. Now, the advantage of converting into polar material is polar materials are more water soluble. And if they are water soluble, they can be very quickly excreted through urine, right? They can pass through the kidney, they get thrown out through glomerular filtration, then get thrown out through urine. That's the purpose of the liver, in one sense, because it converts non-polar molecules into polar molecules. So it quickly gets rid of toxic substances through urine. The other way it does is it also realizes, oh, intestine made a mistake by picking up this molecule. Let me send it back. So now this time when it sends it back, it actually sends it along with bile. You know about bile, the yellow color liquid that's present in your intestine is bile, right? Bile is like a detergent. So now your drug that was picked up inadvertently in the intestine gets mixed with bile, and then it gets thrown out again through intestine. So the liver does both, make sure the molecule gets excreted through urine, also make sure it gets thrown out through the digestive system itself, to ensure that it throws out. Okay. Like I said earlier, all drugs must be excreted from the body. If it is too soon, you will not see any therapeutic effect. If it stays too long, there is toxicity. But you need to achieve some desirable result. You need to have some amount of a half-life for your molecules, and that's again designed by drug design, right? So here I have like a slightly complicated picture of what all can happen in the liver. So liver cells are called hepatocytes, right? Hepatocytes have a lot of transporters on them. Remember, by now the drug is already in blood. Now the liver imports all the molecules that are in blood into the hepatocyte and does that with a lot of transporter molecules, brings it in, and within the hepatocyte, there are lots of enzymes. There are phase 1 enzymes, there are phase 2 enzymes. All these act on the drug molecules that you have. Some of them get thrown back into the bile canaliculi. That means it goes out through bile duct and then gets thrown out through feces. Otherwise, it will metabolize it and circulate it back in blood so that it goes out through urine. So a lot of processes happen in the hepatocyte, and you need to make sure your drug survives all this process. Now, after these, the drug has to finally find the protein that it should bind to, right? And you need to think of what your drug has to do to the protein. So the protein, you need to either inhibit it, that means prevent the protein from doing the job it's supposed to do, or activate it, increase the rate at which that protein is functioning. Or maybe it's a protein that is not functioning, your molecule goes and binds and activates that protein. So either of those functions have to be decided early on. But the most important thing about protein target is you need to design selectivity. There are about at least 30 to 40,000 different proteins in your body. Out of these 30 to 40,000 proteins, there are variants, right? So there are some things called isoforms for proteins. So there can be one protein, multiple forms of that protein. So in reality, the number of different types of proteins in your body can be anywhere upwards of 100,000 to 200,000 proteins. And different people have slightly different amounts of proteins. Different people have something called single nucleotide polymorphism. So their proteins may slightly have certain changes and all that. So all in all, you have at least 100,000 to 200,000 different proteins in different quantities spread across the body. Now, in all this scenario, your drug still has to bind to only one protein. And that's again the job of a drug designer, to carefully design so that it binds to only one. So let me just walk you through now how the drug design process works. It's a bottom-up approach. Now, what do I mean by bottom-up approach is you build the science for it. So you start with a biologist. So the biologist decides what the target protein is. Let's say a biologist comes up and says, hey, we can inhibit this tyrosine kinase. So tyrosine is a common amino acid. The kinase is a protein that adds a phosphate. This person comes to you and says, I'm reading a lot of literature, and the literature says, if we inhibit this specific tyrosine kinase, we may be able to prevent cancer cells from multiplying. That basically it would mean that you can prevent cells from proliferating by inhibiting this tyrosine kinase. So the biologist comes and says, yeah, this is a potential target, right? And once the biologist has thought about, okay, this is the potential target, the biologist has to now design what is called an assay. Now, an assay is an experiment. So you conduct an experiment where if any chemical matter comes in contact with tyrosine kinase, it should be able to bind to tyrosine kinase and prevent tyrosine kinase from doing its job. If it's able to do that, you should see some positive signal. So there are plenty of types of assays. Very common assay are dependent on what is called luminescence or fluorescence. So anytime a protein gets bound to a ligand, gets bound to a drug molecule, there is a change in its shape. And the change in shape can be determined by the change in fluorescence. So you can see that signal output that comes out. So the biologist first is tasked with developing an assay which tells everyone that, yes, if this molecule binds to the protein, then we know that it's able to prevent that drug, it's able to prevent that protein from function. So you need to develop an in vitro assay. Once you develop the in vitro assay, now the question is, what type of chemical matter, what structure of molecules, what type of molecules, what class of molecules are able to bind to tyrosine kinase? So what you need to do is you have to find a source of a huge library of molecules. A lot of drug companies have their own in-house libraries, right? But you don't need to have a full-fledged library of your own. You can also buy libraries from companies. Companies sell hundreds and thousands of drug molecules already dissolved in a buffer and kept in plates. You can buy those plates and you can test every single molecule in that plate for your tyrosine kinase, right? And if you do that and you basically try and see if any class of drugs works, right? So you do the high throughput screen. Many times the number of molecules that are screened for a high throughput screen can actually be as close to 2 million or 3 million molecules, right? Chemists have made a lot of molecules in this world. Even in India, there is a facility in Lucknow where people deposit all the chemistry molecules that they make. It's in a place called CDRI Lucknow, and they basically use those libraries for doing high throughput screens. So if you are a chemistry researcher, you can always deposit your molecules into CDRI Lucknow's library, right? That's how you generate your high throughput screen. So once you complete your high throughput screen, you validate all the positives that you got, and then you say a chemical class we have identified. And that chemical class is called a chemotype. A chemotype is basically a string of atoms in a particular shape that may bind to tyrosine kinase effectively. That's what you would have determined at the end of a high throughput screen. After the high throughput screen comes an important task called a hit identification or lead optimization. There are various names for this. Different companies have different names, different researchers have different names for it, but basically it's a process called hit to lead and lead optimization. Fine. So the aim at this process is to convert something that was just a hit, something that was just a chemotype, into a molecule that can go into a human. That's a big change. A chemotype is just a string of atoms. From there to ultimately become a drug that goes into humans needs a lot of science and a lot of art to go into that molecule, and also a lot of money, right? So if you look at some of the figures that I have here, this number varies a lot. It depends on what type of industry you belong to, what sort of therapeutic area you're in. Oncology may be a little cheap, something like immunology may be very expensive, something like neuroscience can be even more expensive. All in all, from therapeutic concept where the biologist identifies a protein target all the way up to bringing it into humans can take anywhere between 3 to 6 years. It also depends on whether you are the first company in the world to try and do this, or are you following up from someone else. If you're following up from someone else, obviously the process gets a little streamlined and gets faster. But if you're the first company that's doing something like this, it obviously takes much longer. It also takes a lot more money because you have to generate all the scientific data entirely, right? So the estimates vary, but 700 to about 4,000 crores is the usual. That would translate to 700 crores would be like $100 million. 4,000 crores would be like $600-ish million. That's, I think, a rough ballpark to come to a stage where you can get a molecule that is ready to go into human. Once you are ready to go into human, then the cost escalates, because now you will need at least another 4,000 to 5,000 crores, because now you're going to dose into humans. So you're going to have a full-fledged study, which is called clinical trials, go through phase 1, phase 2, phase 3. There are lots of doctors involved, lots of regulatory bodies that are involved, because they're all going into human. I'm not going to touch those aspects today because that's out of the scope of my topic out here. Today's topic will just be the drug design process. So up until going into human is what I'm going to talk about, and that's going to cost you quite a lot, right? Now, you at the outset, you may think, okay, this 700 to 4,000 crores does not seem to be a lot. But the complication here is until and unless you go into human and test it, there is no guarantee that it is going to work. So you could spend like 4,000 crores, go into human and realize there is some toxicology issue in humans, which you did not pick up in all your preclinical work. And then the 4,000 crores just goes waste, right? That's why drug companies have to invest a lot of money into their research process. They also have to have a very robust decision-making process, because you don't want to gamble on a project that is not going to see success, right? Because that's a lot of money to put in. And ultimately, if it's not going to be successful, that's a whole lot of money that goes down the drain. It is also the reason why drugs are expensive. New drugs are expensive. So things like paracetamol have been around for a long period of time. They've been around for a little more than 100 years, paracetamol. That's why you can get a Dolo 650 at like 225 rupees or 330 rupees, depending on the brand that you buy, for 10 to 15 tablets, right? And you buy like 2.5 to 3 grams. You consume about 2.5 to 3 grams without thinking too much, because paracetamol is very, very cheap. But something like a very new cancer therapeutic drug that was, let's say, discovered 2 years ago, came into the market 1 year ago. If you were to think of how much it would cost, each dose would cost you at the very minimum a lakh and a half each dose, right? And you would have to take those doses for a good amount of time. So it's not a one-time dose. So the cost that you are paying basically goes to recover the cost of research and development. So it's a pretty expensive process. It's not cheap in any sense. So the way lead optimization, that is the process of converting a chemotype to a drug, is a process called an iterative design, right? A lot of designing processes around us are iterative designs, but to convert a chemotype into a drug that comes to a market, there are thousands of iterations that the molecule has gone through. Okay, so there are ways to do it. I am not going to go into all the ways that we do it. I'm just going to give you a very brief example. So one of the techniques that we use is called structure-based drug design. These are applicable only for those proteins which you can crystallize and find the structure of the protein, right? There are lots of proteins that are not crystallizable, that do not crystallize. A lot of proteins that are across the membrane, cell membrane, they're called transmembrane proteins. They are very hard to crystallize. They are very large and you cannot crystallize them easily. So for those molecules, if you have to design a drug, you are basically going blind, right? You don't know what the shape of the protein is, and you don't know how your drug is going to fit into that protein. And if you don't have a molecule that fits into the protein, you will not be able to prevent that protein from doing its job, right? You cannot design anything that binds to it. Now you think of a protein as something that has lots of pockets in it. So if your drug goes and binds into one of those pockets, when it binds, the protein's shape will change. And when the protein's shape changes, the protein essentially becomes inactive. A lot of different types of inhibition is there. There's competitive inhibition, non-competitive, allosteric inhibition. Not going to go into those details, but structure-based drug design is a critical tool that any medicinal chemist uses to design drugs. Okay? The whole process, like I said, is iterative. So we have like a process. We design. We say, okay, this is the structure of the protein. I know what's the chemotype that we had. I know how the chemotype sits in my protein. I need to build properties. I need to make sure it crosses the liver, it crosses the intestine. So I have to build those things in it. But I cannot build them at the cost of it not binding to the protein. Ultimately, I can make a drug that's highly absorbed in the intestine, but it may be not potent at all for the drug. So I need to keep my drug's potency first and then design everything around it. So I need to know the structure so that I can build everything around it. So I make a design, I hypothesize, okay, maybe if I put an ethyl group here, maybe it will improve solubility, at the same time maintain action on the protein, right? So I design, and then it's made. The design is executed into an organic molecule that's made by an organic chemist. And then it has to be tested in the assay. Remember, I spoke about an assay where where the biologist tests it. It has to pass that test. And along with that test, a bunch of other tests. I'm going to walk you through that next slide. And then the data that comes out has to be analyzed. Okay, I made this change. Was that change beneficial? It maintained potency, but something else happened. I got something else, but some other issue came up. Now I need to go back, change my hypothesis, redesign, make it again, test it, analyze the data, and make another hypothesis. So I keep going through a lot of design, make, test, analyze cycle, right? And ultimately, this whole process, every time you do the whole process, you will get better and better understanding of the properties of your molecule, and ultimately you will be able to make a drug, right? So I spoke about the assays. We spoke about only one assay, which was for tyrosine kinase. You can make an assay that says molecule is bound to tyrosine kinase, but that's only one part, right? There are hundreds of other kinases in the body, and you need to have a test for all those because you want to make sure your drug binds only to the kinase that you want, but does not touch any other kinase. So you need to have assays in place for all that. So on target, you will have to have an assay, and off target, that's all other assays you need to have, right? The assays can be biochemical. Biochemical is basically a purified protein. You take a purified protein, you put your drug and see if they bind. Or it can be cell-based. The drug permeates into the cell and passes through into the cell, binds to the protein, and you see a downstream effect of that. That's another way to test. It can also be biophysical. So drug binds to protein, protein has a change in its surface behavior, and that can be tested, right? So it can be biochemical, it can be cell-based, it can be biophysical. So those are sort of target and off-target assays that you need to have. See, I cannot take every molecule that I make. I'll make thousands of molecules in the whole process of identifying a drug. I cannot take each of those, give it to a human and say, hey, is it getting absorbed or not? That's a complete waste of resources, and I wouldn't be able to use so many humans to do it, right? So I need to have something that's quick to tell me whether this molecule has a good probability of getting absorbed in the intestine. So I need to have an in vitro absorption test, and that's called a permeability assay. Usually intestinal cells, intestinal cancer cells are grown in a petri dish, and you add your drug and see how much goes into the cells and how much does not go in. The more that goes in means the better it is permeable, right? Similarly, you need to check whether your drugs are resistant to the enzymes in the liver. So you do an in vitro metabolism test. You take the enzymes in the liver, grow them separately in the lab, or you make them separately in the lab. Like put the gene in a bacteria and make that grow, right? So you don't have to take from humans. You can generate those enzymes in a lab. And those enzymes you can now use how much ever you want to test all your compounds. So if your compound is intact on exposure to liver enzymes, that means very low probability that your molecule will get metabolized in the liver, which is good, right? You can have in vitro toxicity tests. Are there other, so there is a very famous story of thalidomide, which is a drug that was put out in the 1960s for morning sickness in pregnant women, right? But ultimately, it was found that thalidomide actually caused genetic disorders in the children that were born. So you need to know whether your molecules are genotoxic, right? Does it cause mutations? So you need to have tests for those. So again, it's done in vitro. You do everything in vitro. You need to do solubility. Does my drug have good solubility? Does my design process, how am I changing solubility? I need to know physicochemical parameters. What's the polarity of my molecule? What's the acidity of it? What's the basicity of it? How polar is it? What's the polar surface area that it has? All these things, you need to have data for all that. So these in vitro assays help you screen thousands of molecules. So thousands of molecules have come from thousands of design, make, test, analyze cycles, right? Because it's in vitro, you can do it. Now, whatever molecule that you design, if it's passed on all these in vitro tests, so if if your designs are a pass for all these tests, then you can take it into animals, right? And the animals that you would first test it on are the really small animals like mice and rats. They're also very similar to humans. Therefore, they're quite apt to do it. So you test them on mice and rats, and then if your drug, is looking good, then you go further to dogs, and then if it looks good, then you go ahead and test it on monkeys, right? So when you're testing on these animals, you are studying what is called pharmacokinetics. Pharmacokinetics is essentially telling you the kinetics of the drugs, like how much is getting absorbed, how long is it staying in the body, how much is getting excreted out, where is it getting excreted, all those things comes up in pharmacokinetics. Pharmacodynamics is basically what the drug does to your body, right? So for paracetamol, the thing that you observe with paracetamol is body temperature comes down as and when you take your paracetamol. That's pharmacodynamics, right? Efficacy is basically curing the disease. So let's say there is a tumor in the intestine. You take the drug and the tumor size reduces. That means that's efficacy, right? And toxicology, as you keep consuming the drug, are you seeing any toxicology? Does the person have reduced appetite? Is the person losing weight? Is he having dizziness? Those sort of things become toxicology, right? So you do the same set of things that you do with humans. You do them with preclinical species, that's in mice, rats, dogs, and monkeys, and you make sure that in all those animals, it is clear that your drug has no problems that shows up on any of these. If all those are fine, then ultimately one molecule from your whole process gets dosed into human. Of course, before it gets dosed into human, there's a lot of regulatory process, right? You have to get approvals from the requisite authorities. You have to give them sufficient data saying this is what you have. You need to give them the profile of everything that you have tested so far. And if the drug authority agrees, yes, everything is looking fine, go ahead and dose it into humans. That's when you can do a first in human trial. And then you go through phase 1, phase 2, and phase 3. Right? So with regard to design, so many papers that talk about drug design usually look like this. Now, let me just move this from the screen. They basically look like this. You see, it has something called a HTS head. I spoke about the high throughput screen head. This was a molecule that came up on the high throughput screen head. It's got, this was a drug that they were trying to develop for hepatitis B virus. When it starts with a potency. Potency is a number that is attributed to talk about how effective your drug is, right? The lower the number, the more potent your molecule is. So it starts at something like 0.3 micromolar. And then a lot of optimization was done, but you can see the core of the molecule where these two nitrogens are, this whole core is still intact, right? The black part is still intact. A lot of changes have been done to other parts of the molecule, like a chlorine and a fluorine, a methyl here, a pyridone, not a pyridone, a morpholine ring here, have all been kind of added. A medicinal chemist has basically gone to work on it and figured out this is the best combination and figured out that ultimately it can go into the human. So you see the potency gets increased by a huge amount from 323 nanomolar or 0.3 micromolar. It comes down to about 0.027 micromolar. This is liver microsome. That means how quickly it gets metabolized in the liver. This number should be as low as possible. This is a pretty high number, 17 and 76. It comes down. Again, telling you that they were able to fix that problem. LISA is a solubility assay that's there. It's a very poor soluble compound to start with, ultimately becomes a highly soluble compound. So a medicinal chemist essentially goes through the whole designing process and ultimately leads to this. A lot of molecules would have been made, hundreds of molecules would have been made. Similarly, another paper has this sort of a thing. Again, you can see the core, which is this nitrogen containing rings in the middle, right? And also this piperidine ring out here. A lot of changes on this side, a lot of changes on this side, ultimately leads to a development candidate. Right? Ultimately leads to a development candidate which comes to a clinic. And if you look at this last one, you see from something which had a potency of 100 micromolar was ultimately brought down to about 0.5. Lots of complicated things were done, lots of docking was done, a lot of microscale synthesis. Microscale synthesis is doing synthesis in really, really small amounts. About 14,000 compounds were apparently made until they finally obtained this molecule, right? So it depends. A lot of drugs can be designed with as little as 200 to 300 iterations. Sometimes when you're doing microscale synthesis or nanoscale synthesis, you have to screen a lot more. You'll have the ability also to screen a lot more. So it can be upwards of 5,000 to 7,000 iterations that you go through, right? Sometimes the way the design is done is hypothesis, and you do clustering, you cluster bomb. You say, I have 5, 6 different hypotheses. I'm going to put everything together. I'm not going to wait for the cycle to complete. I'll put everything together and then do a high throughput synthesis. So you can do that to ultimately reach your molecule. Whatever you do, you would have to go through some amount of work in the design process, right, to ultimately lead you to the drug. For some reason, it's not advancing. So if you ask me at the end what drug design is, it's a lot of data analysis that you need to do. You have to have a good sense of what works and what doesn't work in the iterative cycle. And whenever you see anything does not work, you need to think of the science of how to fix it, how to make it work better. There is an art to it because you need to design to bring balance. Like I said, there are lots of processes. You can make a drug that does only one thing very well, but fails in everything. And that's never going to become a drug for a human, right? It will only become a molecule that was made. It is never going to become a drug. The drug that you make should have balanced properties. And that's where some art comes in. And you need to have a way to make it all work, right? And there is no perfect drug that anyone can make. All drugs will have some issues or the other somewhere, and you need to have a strategy of fixing it. So ultimately, drug design has a lot of data analysis, a lot of science, art in it, and also a lot of strategy. If everything works out, ultimately you will have a drug that will go into human. There are lots of projects that get started and they ultimately never make it to human because certain drugs are simply called undruggable. Sorry, not drugs. Certain proteins are simply called undruggable. So those are sort of proteins where the drug needs to have certain characteristics which will allow it to inhibit the protein, but will not allow it to survive anywhere else in the body. It may not get picked up in the intestine. It may not survive the liver, all those sort of things. So yeah, there are projects that don't specifically take off, will not yield a drug ultimately. So it's an expensive process. It's a low probability of going all the way to clinic and a low probability of finding itself in the market. That's why drugs get very, very expensive. So I'll stop with that. Come close to an hour. I will take any questions that you may have. Thank you. Thank you for giving me this opportunity.
Thank you, sir. Any questions? You can unmute and ask the questions. Jyoti ma'am, any questions from your side?
Yes, ma'am.
Okay, go ahead.
So one of my student is asking question.
Please go ahead.
How to use nanoparticles as drug delivery for a particular tissue? What is the impact on other tissues?
Okay. So nanoparticles themselves may or may not be drugs, right? there are some metal complexes that are made into nanoparticles that could be used as a drug. Currently, there is nothing that's in the market which has nanoparticles that could be used as drugs, right? Like I said, a nanoparticle has to survive a lot of things, which is get absorbed in the intestine, survive the liver, then get circulated, find the protein, and then. So there is nothing out there yet. A lot of it is experimental. People do all those things in the lab. And when you do it in the lab, you can have a lot of control over how you do it. So yes, there is some promise, but it needs to be seen in the future whether any of those get realized into play. Yes. So right now, the kind of effects that it will have, we won't be able to predict anything about the effects that it will have until and unless something starts coming up in human beings.
Sir, one more question.
Yeah.
Is there any transformations in drugs and their delivery process for their therapeutic outcome?
I didn't understand the question. Can you repeat that?
Is there any transformations in drugs and their delivery process for their therapeutic outcome?
Is there any transformation in the drug? Is that
In drug?
what- Yeah. Okay. So sometimes that's a good question actually. So sometimes what happens, like I told you, some drugs become hard to make them get absorbed in the intestine. So what people do is they make a molecule and they attach another piece to it. Okay. So the piece that they attach to it is called a prodrug moiety. So the prodrug moiety gets attached to the primary molecule, and then it's administered into it. And once it passes through the stomach and goes into the intestine, it gets absorbed. And then once it comes into the blood, the prodrug gets separated out from the parent molecule. So the prodrug leaves, and now you have the parent molecule. So ultimately, what you will be doing is with a prodrug, you will be making sure drugs that were otherwise not, you were not able to dose into a living organism, you will now be able to dose. So those are called prodrugs. So yes, they are a way to transform the drug so that you can ultimately dose them. Good question. Yes. So I'm getting a question on the chat. Prodrug means conformers? No. So prodrug is another small chemical entity that is attached to the parent drug. So usually it's things like amino acids that are attached. So when you attach them, the body has enzymes that will cleave amino acids. So let's say you take your drug and you attach an amino acid to it, which helps the drug to get absorbed in the intestine, then goes into blood, and then these enzymes come in and cleave off that amino acid. So now your parent drug is available in the blood, and the amino acid gets processed. So no, it's not a conformer. It's a separate chemical entity that's attached to the parent drug. How to do solubility test? Okay, so it's very, very straightforward. You take the solid, which is your drug, add buffer. Okay, so you don't want to do, there's no point in doing solubility in water because inside your body, everything is pH controlled. So everything has to be in buffer. So you get something called a simulated intestinal fluid. It's an external solution which has some bile in it, and it's maintained at either pH 6.5 or at pH 5 based on whether you're fasting or your consumed food pH changes. So two different pHs, and it's called a simulated intestinal fluid. So take a vial, put your drug molecule in it, add this simulated intestinal fluid, cap it, shake it. After a while, allow it to settle down, and then check whether any solid is left. Okay. Now, this is a very crude way to do it. It is not going to be quantitative, what I just explained. What I explained is very qualitative. But the quantitative way of doing it is people do HPLC. I don't know if you have heard of HPLC. So they take the liquid and they do an HPLC and see what is the concentration of the drug that they have got. From that, they can back calculate and decide how much solubility they have. I see. What is your opinion on docking studies that are carried out using some computer software? Yeah, like I said, we use docking extensively. It's a very important tool. It has a specific purpose. If you know the structure, it can be very, very useful to have docking, because it tells you what designs will work and what designs will not work for the protein, right? Suppose you have a hydroxy group and you convert that into a methoxy group. Is that tolerated by the protein? Can be very easily answered by docking. And if the docking studies say, no, it is not going to be tolerated, then you don't have to make it, right? So docking studies are very important for the drug design process. But it's not the end of the drug design process. It's only one of the tools. It's only one small contributor. There are other things also that are required. Are there how to change physicochemical properties of the drug? Yes, like what I said just now. Like if you have a hydroxy group, that's a polar group. So hydroxy has a hydrogen that can be donated. So it's a hydrogen bond donor. So if you convert that hydroxy group to a methoxy group, which is OCH3, then there is no more hydrogen bond. So simple changes to the molecule can change its physicochemical properties. Sometimes you will have a benzene ring, you change that benzene ring to a benzene ring that has a nitrogen atom, like a pyridine ring. And that will again change the physicochemical properties. So it's quite easy to change the physicochemical properties. It is harder to decide what physicochemical property you need. So it's easy to change. It's harder to decide what you need. So that's the more complicated. Any other questions? Okay, what is the meaning of bioavailability of the drug? So the actual meaning is slightly more complicated, but I'll give you a simple idea of what bioavailability means. So you take the drug and you consume it orally, right? So when you consume it orally, some of the drug is going to get absorbed in the intestine and some of it is not going to get absorbed. It's only whatever that gets absorbed in the intestine flows through the blood. And so you measure how much drug comes in to your blood. So you take blood samples and you check how much drug is present in that. Fine. If a drug has low bioavailability, then the concentration of the drug in blood will be low. If a drug has high bioavailability, then the concentration of the drug in blood will be higher. So it's kind of roughly what it means. It is not the exact way. The exact way to do it is actually what is called measuring the area under the curve. So you need to do an IV dose, then you have to do a PO dose. You have to take the AUCs for those and divide them. But for us, what it would mean simply is low bioavailability means less drug is absorbed. High bioavailability means more drug is absorbed in the intestine. I hope that answers the question. Any other questions?
Chat box, one more question. Okay, there, thank you.
Yes. Yeah. I'm happy
Sir,
to do it.
Sir, one more question.
Yeah.
Usually there is a practice of taking half of the tablet. Just give some clarification.
Yeah. I mean, so usually nobody so if you do it, nobody actually recommends half a tablet. But it's fine for simple things like for paracetamol, there is no problem. For a capsule, you cannot take half. Okay. A capsule is just a tube, and if you tilt it one way, all the molecules are on one side. If you tilt it the other way, all the molecules are on the other side. So it is for simple things, it's fine. For paracetamol, no problem. For things that are that have things like sustained release or extended release, those sort of things, when you break the tablet, you break the coating on the surface. So it's the coating on the surface of the tablet actually that makes the tablet release the drug slowly. And for some people, it will be required. For some people, it may not be desired. So for those sort of molecules, for those sort of drugs, you should not break the tablet into half. For capsules, do not break them into half. For very simple things like Dolo 650, sure. For things like ibuprofen, sure, not a problem. For things that are specialized, you should not be doing half tablets. Yeah? And the problem, of course, is the fact that after you break the tablet into half, you are exposing the side of the tablet that should not be exposed to air. So there may be some degradation of the drug molecule. So yeah, I wouldn't do it, but for paracetamol, it should be okay. For Dolo 650, it should be fine. I see a couple of more questions.
Yes, sir.
Yeah, I'll go ahead and answer them. So is cloning technique helpful for drug design? Yes. For all the in vitro assays that are done, right? Because it's done outside of the living organism, you have to clone. There is no other way to do it. Let's say there is a protein in human, tyrosine kinase. This tyrosine kinase, you cannot take 50 human beings, take tyrosine kinase from them and purify the protein. Right? You cannot do that. What you will do, however, is take the genetic sequence for tyrosine kinase from humans. Then take that genetic sequence, clone it into a bacteria, make that bacteria grow, then make that protein production in the bacteria, then isolate the protein and use it. So yes, if there was no cloning, a lot of the drug assays would not be there. So drug designing would have been a very complicated process if there was no cloning technique. The other question that's there here is what part of the drug binds to the chemoreceptor on the enzyme? It depends. So some drugs have only one point which is recognized by the enzyme. Some drugs have multiple points. There are drugs nowadays that recognizes two enzymes at the same time. That's also possible. So you have one end of the molecule which binds to one enzyme. There is a big linker in between, and then another end which binds to another enzyme. So you can inhibit multiple enzymes at the same time. So for the one that we spoke about, tyrosine kinase, kinase is also an enzyme whose job is to phosphorylate. Kinases have a region called the hinge, and most molecules that are designed for kinases actually bind in the hinge region. Again, this is only for kinases. For different proteins, there will be different regions. So there is no universal place where the drug binds. The drug can bind to the active center, in which case it's called a competitive inhibitor. That means it's competing with the substrate for the enzyme. Or it can bind in other parts of the enzyme, in which case it will be called non-competitive. So there is no fixed part where it should bind. But for certain classes of proteins, there are some fixed points. But there's no general answer to it.
What is the role of X-ray crystallography?
Oh yes, yeah. X-ray crystallography is super critical, right?
What is the role of X-ray crystallography in physicists?
Oh, so yes, physicists who do X-ray crystallography work are usually employed in drug discovery companies. You remember I told you that there is structure-based drug design where the drug binds to the protein. So, which means somebody has to find out where the drug binds to the protein. Now, you cannot see it, right? A protein is just another powder, a drug is just another powder. You just mix the two, you cannot see anything. The way you need to fix what is the shape of the drug molecule, where does it bind to the protein, is all done by X-ray crystallography, right? So X-ray crystallography can be multiple types. You can have single molecule X-ray crystallography in which only the drug molecule's shape is found. That also is done. Drug molecule co-crystallized with the protein is where it's even more important. So wherever it's co-crystallized with the protein, you cannot just do X-ray crystallography in a lab. It's not in a normal lab, you cannot do. You need a much more powerful X-ray beam. And that much more powerful X-ray beam usually comes from a synchrotron, which is a very fast particle accelerator and produces very high energy X-rays. And that has to be used. So sometimes what people do in academia is they send X-ray, the drug crystallized with the protein, they send it to Europe. And you know, this Large Hadron Collider in Europe is one of the largest particle accelerators. They have lots of places where they harvest the X-rays that come out of this particle accelerator. And that is powerful enough to help physics people determine the shape of the protein and how the drug is bound to the protein. So yes, it's absolutely useful to do X-ray crystallography.
Sir, one question.
ಹೇಳಿ.
ಈಗ ಗ್ಯಾಸ್ಟ್ರಿಕ್ ಅಂತ ಟ್ಯಾಬ್ಲೆಟ್ಸ್ ಹಾಕೋತಾರಲ್ಲ ಯಾವಾಗ್ಲೂನು. ಆಕ್ಚುಲಿ ನಾನು ಪ್ಯಾಂಟೋಡಾಕ್ DSR ಯೂಸ್ ಮಾಡ್ತೀನಿ, ಅದು ಕ್ಯಾಪ್ಸೂಲ್ ತರ ಇರುತ್ತೆ, ಬಳಸಬಹುದಾ?
I mean, I'm not a doctor, so
ಅಂದ್ರೆ ಅದು ಡ್ರಗ್ ಅಷ್ಟು ಯಾವಾಗ್ಲೂ ಬಳಸಿದ್ರೆ ಏನು ಅದು ಡ್ರಗ್ ಇಲ್ಲ, ಏನಕ್ಕೆ ಅದು ಗ್ಯಾಸ್ಟ್ರಿಕ್ ಅಂತ ಕೊಟ್ಟಿರ್ತಾರೆ, ಅದ್ರ ಸೈಡ್ ಎಫೆಕ್ಟ್ಸ್ ಏನು ಇರಲ್ವಾ ಅಂತ?
ಇರತ್ತೆ. Like I said, there is no drug without any side effects. There will be. So adana manage ಮಾಡಬೇಕು. So the best person to talk to will be your doctor, and without doctor's consultation it is not recommended that you take.
ಅದು ಕೊಟ್ಟಿದ್ರು, ಡಾಕ್ಟ್ರೇ ಕೊಟ್ಟಿದ್ದು.
But usually ಅವ್ರು ಹೇಳಿರ್ತಾರೆ ಎಷ್ಟು ದಿನ ತಗೋಬಹುದು.
ಹೇಳಿದ್ರು ಫಸ್ಟ್ ಒಂದಷ್ಟು ದಿನ ಅಂತ. ಈಗ ಯಾವಾಗ್ಲಾದ್ರೂ ಸಮಸ್ಯೆ ಆದಾಗ ತಗೊಂತೀನಿ, ಅದು ಕೇಳ್ಬೇಕಾ ಡಾಕ್ಟರ್ ಹತ್ರ?
ಐಡಿಯಲಿ ಕೇಳಬೇಕು ಡಾಕ್ಟರ್.
ಓಕೆ ಸರ್,
ಯಾಕಂದ್ರೆ ಸೊ ನೀವು ಮೆಡಿಸಿನ್ಸ್ ತಗೊಂಡಾಗ ಬೇರೆ ಯಾವ ಮೆಡಿಸಿನ್ ತಗೋತಾ ಇದ್ದೀರಾ ಅದ್ರ ಜೊತೆಗೆ ಅದು ಆಲ್ಸೋ ಬಿಕಮ್ಸ್ ಇಂಪಾರ್ಟೆಂಟ್. ಸೊ one drug interacts with another drug, so it's called drug interaction. So those things also become important. So usually the best person to recommend what is the right dose, what is the right time period to take should be your doctor.
Okay sir, thank you.
Yes
Sir, follow-upping this, there is always one gastric tablet before painkillers and all. What is the thing? Yake?
So usually a lot of the drugs stimulate your stomach to produce the stomach enzymes, which are also acidic, right? So there are gastric stimulation agutte ನೀವು ಡ್ರಗ್ ತಗೊಂಡಾಗ. ಸೊ ಡ್ರಗ್ ತಗೊಂಡಾಗ ಗ್ಯಾಸ್ಟ್ರಿಕ್ ಸ್ಟಿಮ್ಯುಲೇಷನ್ ಆದಾಗ ಇವಾಗ ಬರೀ ಹೊಟ್ಟೆನಲ್ಲಿ ತಿಂದ್ರೆ ಅದು, if there is not enough food, then it will start damaging your gastric wall. So ಅದಕ್ಕಿಂತ ಮುಂಚೆ ನೀವು ಗ್ಯಾಸ್ಟ್ರಿಕ್ ಟ್ಯಾಬ್ಲೆಟ್ ತಗೊಂಡ್ರೆ ಆ ರೆಸ್ಪಾನ್ಸ್ ಸಬ್ಡ್ಯೂ ಆಗ್ಬಿಡುತ್ತೆ. So the stomach does not react to the drug as much as it would otherwise react. So many times the drug ತಗೊಳ್ಳುವಾಗ you will also take a tablet that will control the gastric response. That's the reason to do it.
Thank you, sir. Any more questions? I think we can end here. We are running out of the time.
Yes. Thank you.
Yes, sir.
Thank you very much.
I could like to extend our sincere thanks to our resource person. Thank you, Kiran sir. The presentation was not only informative but also engaging, making complex world of design more accessible to all of us. We are truly fortunate to have you here. I also thank all the participants for active participation and thoughtful questions. Once again, thank you one and all. Thank you, sir.
Thank you.